Hello, my name is Sophia Fagan, and today is January 7th, 2020. I will be talking to you today about the digestive system and the enteric nervous system that is involved. Today, I will be talking to you about some features of the digestive system. The enteric nervous system is a division of the autonomic nervous system of the body. This nervous system functions for the control of the gastrointestinal tract. The enteric nervous system can act independently. This means that it can carry out a reflex independently of control by the central nervous system. Therefore, the enteric nervous system can receive a stimulus and then act on this stimulus. The enteric nervous system controls the motility, secretion, and growth of the digestive tract itself. As you can see on the image on the right, this is a depiction of the different parts of the gastrointestinal tract. The enteric nervous system will control the function of this entity as a whole. The enteric nervous system shares many functions with the central nervous system that we have previously learned about. These similarities are similar anatomically and functionally. These include intrinsic neurons, which are the neurons that lie completely within the walls of the gut, in the same way that the interneurons are contained in the central nervous system. Similarly, the neurons of the enteric nervous system release neurotransmitters and neuromodulators that are identical to the molecules found in the brain. As you can see in the structure on the right, one of the best-known gastrointestinal neurotransmitters is serotonin. The enteric nervous system also has glial cells of the neurons. Additionally, the capillaries that surround the ganglia in the enteric nervous system create a diffusion barrier like that of the cerebral barriers. Lastly, the reflexes that originate in the gastrointestinal tract can be integrated and act on without these neural signals ever having to leave the enteric nervous system. This is why the enteric nervous system is its own integrating center. In the gastrointestinal tract, we have two types of reflexes that can occur. These two types of reflexes are quite different. There are short reflexes and long reflexes, and we will go into a bit more detail about each of them now. First, we will start by discussing what short reflexes are. The enteric nerve plexus in the brain allows the enteric nervous system to act as what many refer to as a little brain. This allows reflexes that are local to begin in the gastrointestinal tract, be integrated in the gastrointestinal tract, and then end in the gastrointestinal tract, where everything is taking place in the gastrointestinal tract. Short reflexes refer to the fact that the reflex originates in the enteric nervous system and then is integrated there without the input from any outside sources. In the depiction of a short reflex on the right, you can see that the sensory receptor picks up the signal of a sensation, brings it to a sensory cell synapse in the gastrointestinal tract, and then generates a response, all happening within the gastrointestinal tract. Now, we will discuss long reflexes, which are different from the previous short reflexes that we just discussed. No matter where they originate, digestive reflexes that integrate in the central nervous system are always referred to as long reflexes. The long reflexes that originate outside of the digestive system may include feed-forward reflexes and emotional reflexes, which are called cephalic reflexes, given the fact that they originate in the brain. They begin with such stimuli such as sights, smells, sound, or the thought of food, and then they act by preparing the digestive system for food that the brain is anticipating. An example of an emotional response that affects the gastrointestinal response is the concept of butterflies in the stomach. During long reflexes, the smooth muscles and glands of the gastrointestinal tract are under autonomic control. It is generally thought that the parasympathetic division is excitatory and enhances gastrointestinal functions, which leads to the nickname for this division being rest and digest. On the other hand, sympathetic neurons generally inhibit the normal function of the gastrointestinal tract. In the depiction on the right, you see the pathway of a long reflex. It begins with the sensation that is sensed by a receptor cell. This carries it to the sensory cell synapses in the spinal cord or brain, where it is then integrated. Then, the response is carried to the peripheral ganglion and to the gastrointestinal tract. This is a long reflex. In this diagram, found in the Silverthorne textbook, you can see the integrations of various digestive reflexes. You can see the long reflexes indicated by the gray arrows, and the short reflexes indicated by the red arrows. Remember that long reflexes are integrated in the central nervous system. Some long reflexes can originate outside of the gastrointestinal tract, but others originate in the enteric nervous system. It all depends on where they are integrated. The short reflexes, remember, originate in the enteric nervous system and are carried out entirely within the wall of the gut. You may notice that some of the digestive reflexes can be integrated by both the long and short reflexes. For example, the distension, presence of food, osmolarity, and acid may all trigger long and short reflexes to the sensory receptors and neurons. You notice that the gray box indicates the part of the enteric nervous system, also known as the little brain. This includes the sensory receptors and neurons, the interneurons, and the enteric neurons. One pathway that only involves long reflexes, as seen here, is the cephalic phase of digestion, which is a feed-forward response triggered by things like sight or smell. These are sensed by the sensory receptors and transferred to the cephalic brain, and then by the sympathetic and parasympathetic neurons to the enteric neurons. You can also see that both long reflexes and short reflexes can involve smooth muscles and exocrine cells, and they both can also change the motility of the GI tract and can cause the release and synthesis of compounds.